friends, my name is Nikita Bhode and today we are going to discuss about the genetic engineering. The genetic engineering, also called as a modified process that used to laboratory based technologies to alter the DNA makeup of an organism. This is include the changing is a single base pair, that is a T and or a C G, deleting a region of a DNA or adding a new segment of DNA. This called as a genetic engineering. Means into the DNA fragment, segment, one portion is evacuated and a second DNA's little part is get added in that first DNA. That engineering is called as a DNA engineering. Genetic engineering. Genetic engineering may include the adding of a gene from one species to the organism from a different species to produce a desired trait. Used in a research and industry, genetic engineering has been applied to the production of cancer therapies, brownies yeast, and the genetically modified plants and livestock. Genetic engineering has changed over the years from the cloning of analysis laboratory are used to truly synthesize biology for the understanding and a new biometric capabilities. Genetic engineering, the artificial manipulation of modification and recombination of DNA or other nucleic acid molecules in the order to modify an organism or the population of organism. The term genetic engineering is generally used to refer the methods and recombinant DNA technologies which emerged from basic research in a microbial genetic engineering is generally used to refer the method of recombination of DNA and recombination of DNA technology which emerged from a basic research in a microbial genetics. The techniques employed in a genetic engineering have led to production of medically important products including the humans. Insulin, human growth hormone and the hepatitis B vaccine as well as the development of genetically modified organisms such as disease resistant plants. That is, genetic engineering is a direct modification of an organism genomes, which is a list of specific trait genes stored in a DNA. Changing of genome enable engineering to give the desired properties of different organisms. Organisms created by the genetic engineering are called as genetically modified organism, that is GMOs. Genetic engineering can be done in a variant of the ways, that is manipulation of DNA, cell transplant, transgenics and the cloning. After that, the stages of genetic engineering. First one is a DNA cleavage, second one is a recombinant production, third one is a cleaning and the last one is a screening. In first stage of genetic engineering, DNA cleavage. The DNA cleavage restriction endonuclease cleaves the DNA into the fragments. Means the restriction endonuclease cleavage DNA into the fragments means the endonuclease cleavage is get break down and divided into the fragments. And after that, the recombinant DNA production, fragments inserted into the vectors. In that step, recombinant of DNA, the fragments are inserted into the vectors. After that, the cloning, more recombinant DNA created. After the DNA fragments are inserted into the vectors, they started to recombinant and another DNA is created. And after that, last one is a screening. Is the most challenging part of any genetic experiment. In that, the preliminary screening of clone is done. After that, the finding of gene interest is done, in that the hybridization and polymerization chain reaction is occurs. In polymerization chain reaction, the denaturation, there are four steps. In that, the denaturation is the first step, the annealing, the parameters, and the primer extension. These are the three stages of a polymerase chain reaction. And southeastern blotting, restriction fragment length polymerization, and DNA fingerprinting. All these steps are involved in a polymerase chain reaction or the screening part. This is the stages of the genetic engineering. That is a genetic engineering, also called as a modification, is a modified process that laboratory based technologies to alter the DNA to make up of an organism. DNA engineering is nothing but the when two plants or two animals has a different qualities and the different strength. When if we want that particular one character in a one person and second character in a second person, that both are emerge, and that means the DNA, both factors DNA is get collected, and that DNA, the small portion of a DNA is evacuated, and the small portion of DNA is get installed in that first DNA, and to form another variety, that is also called as a hybrid variety. In this genetic engineering is also done in a human as well as the plants. If two plants have different characteristics, then two different plants have two different characteristics. We need that both characteristic in a single plant, then we can done the genetic engineering. That means the both in both plants a small portion is get collected and emerge them, and after that the after planting them, that part, the both characteristics are occurs and in a single plant. That is also known as a genetic engineering. Genetic engineering is a direct modification of an organism genome, which is a list of the specific traits, that is genes, stored in a DNA. Changing the genome enables engineering to give desirable properties from the different organism. Organisms created by the genetic engineering are called as a genetically modified organism. Genetic engineering is an artificial manipulation, modification, that and a recombination of DNA, other nucleic acid molecules in order to modify an organism and the population of organism. The term genetic engineering is generally used to refer the methods of recombinant DNA technology, which emerged from a basic research of microbial genetics. The techniques employed in a genetic engineering have led the production of medically important products, including the human insulin, human growth hormone, and the hepatitis B vaccines, as well as to develop the genetically modified organisms such as the disease-resistant plants. Genetic engineering has changed over the years from the cloning for analysts and the laboratory use truly synthesize biological and understanding a new biometrical capabilities. So uh, we uh, we seen the general information about genetic engineering in that video. After that, the variety of genetic engineering can be done in a various of forms. The forms are get studied in that video. After that, the stages of genetic engineering we seen in that particular video. That's it. Thank you so much.